but we're in no hurry.
There's no one in
Is there anything that's being omitted from chapter 17? The answer is no. It would be at the bottom of this slide right there. Nothing being omitted. Chapter 17. You can wish.
You just remember the whole chapter.
This is a short chapter without a lot of detail. So enjoy chapter 17. Just some information that usually people find very interesting about hormones. Because this chapter's all you want to know about hormones without using names. We'll do that in chapter 18. Chapter 18 talks about, it seems like dozens of hormones. And a lot of the hormones we've covered in detail in the chapter, and we're not going to cover it, but you're still responsible. But you should already know it. What does ADH do? Yes. And where is it secreted from? Yeah, you know these things. So. When is it secreted? No. When is it secreted? What's the trigger? Not, that is a stimulus, but not usually the one that triggers it first. Indirectly. It's increased osmolarity of the interstitial fluid around neurons in the supraoptic nucleus of the brain.
Yeah, that's what I was
I
gonna say.
thought. That's what you were gonna say? Okay. You were just giving me the abbreviated version?
Exactly.
Nice try. Okay, so let's go and continue on looking at hormones. Again, this is very important information for when we get to chapter 18, and it helps you understand a lot how hormones work. So we're going to continue on with the characteristics of hormones. First of all, there are 3 general characteristics of hormones, and there's 2 on this slide. One is explained in more detail on the next slide. It doesn't fit on here. Stability. All hormones have to be stable in the blood for a while. Some last longer than the other, but they got to be put into the bloodstream, the definition of hormone. And then they have to persist for a while so they can produce their effect. Some last for minutes, others last for hours or days in the bloodstream. Communication. Hormones have to interact and to stimulate certain cells, the target cells. And those target cells have to have receptors for that hormone. The reason they're target cells and they're affected by the hormone is they have a receptor for it. Other cells in your body that don't have a receptor aren't targets of it. Which hormone do you think has the largest number of target cells in your body? Insulin would be my first guess too. Yeah, because virtually everything's that neurons responds to it. Okay? So understand those cells to respond have to have a receptor for that hormone. Now, the third characteristic, a general characteristic of hormones, is that they must be distributed throughout the body in the bloodstream to get to the tissues. They have to go through the bloodstream. Now, there are 2 classes of hormones. I think I mentioned them already. Did I? Is it in the outline above this? Or is this the first time I'm going to say the 2 classes? This is the first time? Okay, because I didn't look what we had already covered. Never mind, I'll tell you anyway. There are 2 classes of hormones. A large number of hormones are considered water soluble, and you know what that means. The other are lipid soluble, okay? Those 2 hormones have different ways of getting carried in the bloodstream. One's soluble, water soluble obviously, and one's not soluble, lipid soluble. Water soluble hormones can float free. They can. Sometimes they're carried by other proteins, but they can float free in the bloodstream. They typically have a shorter half-life. And the reason is because there are water-soluble enzymes in the plasma to degrade molecules that aren't supposed to be there. So water-soluble hormones typically have a shorter half-life, okay? Lipid-soluble hormones have to be carried by what are called binding proteins, and we talked about those in chapter 19 when we mentioned albumins and globulins were sometimes transport proteins. That's some of the things that have to be carried, okay? Can you think of a lipid-soluble hormone? Let's see if you can think of one that you already know is lipid-soluble.
Testosterone.
Testosterone. That hormone is made from cholesterol, and you know that's a lipid. Okay, they typically, because they're lipid-soluble, they're not acted on by those water-soluble enzymes that would degrade a water-soluble hormone, okay? They're carried by binding proteins, and as a result, not being subject to these plasma enzymes, they persist in the bloodstream, typically long. Okay? And those binding proteins do protect them from degradation. Now, about the lipid-soluble hormones. Most of the lipid-soluble hormones are carried by the binding proteins. When they're attached to that binding protein, they're not able to produce a response. Only the lipid-soluble hormone that's not bound to it, and that's only a small fraction of them, those are called free hormone molecules, and they're able to produce a response. So understand there's a huge amount of lipid-soluble hormones in the bloodstream. A vast majority of it, depends on the hormone, is carried by the binding protein. They can't produce a response, but the free ones can. And there's not many free. Yes?
Can you give examples of each type?
Well, a water-soluble hormone, growth hormone. There's
What
one.
about the lipid-soluble ones, the ones that are, can be free-floating and?
Oh, every lipid-soluble hormone is carried by a binding protein. Only a small fraction of it is free. So they bind to a binding protein, jump off for a while, go bind to another binding protein, and they do that. So there's only a small fraction that's actually free in the water-soluble phase of the plasma, okay?
Is there something that causes it to become free and away from the protein, or
No, they
it just
just-
does it have?
They bind for a while and just come loose. Yeah. Okay? Remember, only the free lipid-soluble hormones, not bound to the binding protein, can actually produce a response. So if you look at, for example, testosterone in a male, there's a small fraction of it free and a huge reservoir of one bound to the binding protein. The thyroid hormones are made from amino acid tyrosine, and they are lipid soluble. So thyroid hormones, T3, T4, are lipid soluble, and they're mostly bound to the binding protein. If you remove someone's thyroid gland, immediately no more thyroid hormones are going to be produced. But it takes a week or more before they show the effects of not enough thyroid hormones, because there's all this reservoir that's present in the blood. Now there's only a small fraction that's being used, and gradually there's less and less of the bound protein. So they can go a week or more without a thyroid gland and not show any effects of losing the thyroid hormones until a week later, because there's a reservoir of the hormone on the binding protein. Okay? Whereas water-soluble hormones, every molecule, now some are carried part of the time by a binding protein, but the vast majority of all water-soluble hormones are free and available to produce a response at a targeted time. Okay, everybody understand the difference between these two major classes of hormones? Very important to understand. Okay, and you see their classes determine a lot of their properties. The chemical nature of hormones, again, there's two classes, lipid soluble and water soluble. Lipid soluble hormones include all the steroid hormones, aldosterone, cortisone, cortisol, did I say aldosterone? Testosterone, estrogen, progesterone. Those are the major ones of the steroid hormones. Thyroid hormones, as I mentioned, were lipid soluble, lipid soluble. And there's another class of hormones called the chysanoids, which is poorly studied, and there's not many examples of it. And they're produced from fatty acid, and again would be lipid soluble. Now, they exist in the bloodstream for a long time, but when those free lipid soluble hormones go through the liver, it goes and attaches a water soluble molecule to it, making the lipid soluble hormones then be able to be degraded, okay, and removed. So conjugation is attaching that water soluble component to the lipid soluble hormones that make them more susceptible to degradation, okay? If you guys in the back want to move up close, you can. We have a fairly empty room today. It's like this every Thanksgiving when I give a lecture. Okay, now the water-soluble hormones come in three classes. We have proteins, and these are large amino acid chains. Peptides are short amino acid chains. So growth hormone would be a protein. Insulin would be a peptide, short amino acid chains. And then there are amino acid derivatives where they take one of the 22 or so amino acids and alter their structure. So 21 amino acids, 21. And they alter the structure slightly, put a small group here and there, and it becomes a hormone. Now, of the water-soluble hormones, some are free molecules, some are held bound, okay? And the ones that are floating free and they're small, like the peptides, amino acid derivatives, are removed quickly. I mean, the amino acid derivatives are the size of amino acid. They, of course, go into the filtrate, and unless they're recovered, are going to be excreted. So, in general, water-soluble hormones, again, have a smaller half-life. Ones that attach to a large protein, of course, will not go into the filtrate. They will persist in the bloodstream for a longer time. Whenever a water-soluble hormone binds to a receptor, after it is bound to a receptor, produced the response in the target cell, then that protein and the bound hormone, the receptor protein and the bound hormone, are brought inside the cell and degraded by a lysozyme. Okay? They just break them down to amino acids, or depends on what it is, but they break it down. So water-soluble hormones bind to a cell, produce a response, and then most often they're going to be destroyed. Some water-soluble hormones can be stabilized, increase their half-life by attaching a carbohydrate to it, especially these proteins. So if a carbohydrate is attached to it, converting the protein to a what? What's it called? Glycoprotein. So attaching a carbohydrate to those protein hormones increases their half-life, and they persist for longer. And as a glycoprotein, they can still produce a response in the target cell. Okay? So that's a little bit more about the chemical nature. We're going to see, like when we talk about how they produce an effect in a cell, water-soluble and lipid-soluble have different methods of producing a response in the target cell. Lipid-soluble hormones, can they go through the cell membrane? Yes. Water-soluble, they can't. How do they produce a response? They're both going to bind a receptor. One's inside the cell, because the lipid-soluble can go inside, and one is outside the cell, in the plasma membrane. You see these two classes have a lot of different properties. Which one persists in the bloodstream more? Lipid-soluble. Which ones have a big reservoir? lipid soluble. Yeah. Which ones would have the shorter half-life and be able to be turned over? I mean, you want to express or produce a response and then not have it last very long, you would have water soluble. If you want to control something like reproduction, would you want something that lasts a long time or something that lasts a short time? Long time. So you'd see most of those would be lipid soluble. So there's a lot of different properties and different uses for these hormones. Now, patterns of hormone secretion. There's three kinds of patterns of hormone secretion. They're fairly straightforward. One is chronic hormone secretion, where it varies around a mean or an average over time. It goes up and down a little bit. What do you think controls the level of a hormone in that case? Negative feedback. Yeah, so negative feedback is gonna, if it ever gets too high, produce less, if it ever gets too low, produce some more, and it varies around a mean. Can you think of a hormone that would be that way? Glucagon? Nope.
Is it insulin?
Insulin? Nope. That varies from really high levels to really low levels.
Lutein hormone, FSH,
Not, no.
TSH.
No, okay. She said two reproductive hormones, LH and FSH. In a female, definitely not constant. In a male, it would be. So testosterone is very constant, LH is very constant, and FSH is very constant in a male. Whereas in a female, not so. Okay?
What about parathyroid?
Parathyroid hormone would be, no. Every time your blood calcium goes up, little there. If it ever happens to go down, then it's produced. So it would be a different production. Acute hormone secretion cause, there is irregular productions of the hormone. So some stimulus causes it to be produced a lot, and then a little, and then a little more. What would that be? That could be parathyroid hormone.
Or insulin?
Oh, I'm definitely insulin, yeah. Okay, this one has periodic, not regular amounts of hormone being stimulated and produced. There's a lot of hormones that are done by this acute. There's irregular in time and irregular in amount. So if your calcium level drops just a little, you get a little produced. If it drops more, you'll get more produced. Okay? Everybody understand that one? And the last one is episodic. They happen at periodic times. Female reproductive system, yeah, we've got pulses of hormones being produced over time, and they're regular.
Is it also growth hormone?
Growth hormone is produced at a very constant level. But there can be things that cause it to be produced in more. Okay, control of hormone secretion. What controls the production of any of those hormones? There's three stimuli that can cause a hormone to be produced. And the same three stimuli can limit or hinder, prevent hormone release. Okay? So I'm going to present them as these three things can stimulate a hormone being released, but they're the exact same things that can inhibit or prevent a hormone from being released. The first one is, and the names are going to be confusing, so bear with it, we're in the names. Humoral. This means something in some liquid stimulates the production of a hormone. So here we have low blood calcium. So, and we're looking at an endocrine cell in the parathyroid gland. Now, we have low blood calcium in the bloodstream. Does that directly affect it, that cell? The answer is no. This cell is not exposed to the blood. period. It's not it's not in the bloodstream, it's not in the wall, it's outside of it. So we have low blood calcium, what directly affects the endocrine cell? The interstitial fluid. Don't make it too hard here, okay? So we know that ions can leak out, and so there's an equilibrium between the amount of calcium in the bloodstream and the amount of calcium in the interstitial fluid around these parathyroid hormone cells. So the amount of calcium in that interstitial fluid is what this cell is exposed to. When the calcium levels drop below a certain level, it secretes parathyroid hormone, which is going to stimulate osteoclast, release some calcium. This is one of its effects, and cause the blood calcium levels to go up. Okay, so when you get a blood test, they're measuring this. But as far as parathyroid hormone, this cell is exposed to the interstitial fluid, which is in equilibrium, but I like to be precise in my language, and I want you to be precise also and understand exactly how these work. Can you think of another cell, another hormone that's released with a humoral signal?
ADH?
ADH, correct. Got it? Because it's measuring the osmolarity of the interstitial fluid, and I've emphasized that, around it. Very good. Everybody got that? Those are examples of hormones that are released by a cell in response to some fluid, and it's almost always going to be the interstitial fluid around it. Okay, the next signal is neural control. Here we have a neuron cause the endocrine cell to release its hormone, okay? The adrenal gland medulla cells, these cells are called chromaffin cells, is one of the names for them, and an action potential going down a neuron, it's not myelinated, in the sympathetic division, it should be myelinated, would go and stimulate these cells. When it releases the neurotransmitter, would cause them to release it. Now, they're showing this one over here as inhibiting it, and this one is stimulating it. We don't have that in the adrenal medulla. We only have the stimulatory one. No stimulation, no hormone release. Okay? They're showing the inhibiting on the same slide, but we're not going to discuss the inhibiting. There's not too many hormones that are released like this. Can you think of another one where a neuron tells it to be released? Aldosterone. Aldosterone is secreted in response to humoral signals. And the next one, a hormonal signal. Angiotensin's a hormone. Can you think of another one? I'm trying to think, I guess the pineal gland would be one. And it releases melatonin in response to another neuron. There's not too many, like I said. Okay? So this is the major one. Everybody understand this? It's a neural signal, a neuron, action potential, release a neurotransmitter, the cell secretes. The last one is a hormonal signal. Now, don't let the diagram confuse you yet. Let's look at the definition. One hormone causes the release of another hormone. Okay? One hormone causes the release of another hormone. So here we have a target endocrine cell. It's going to release some hormone that's going to target that cell. Don't get lost in the diagram yet. So this endocrine cell is going to release a hormone, right here, these little triangles, that cause the target cell to change its response. Got it? This is a hormone. This hormone is stimulated by the release of another hormone. So In this case, the anterior pituitary would release a hormone that would cause this target cell to release its hormone. But it can get more complex than that. The hypothalamus can release a hormone that causes the anterior pituitary to release its hormone, which causes the target cell to release its hormone. This sequence of events is very common. So we have and a hormone that causes the release of another hormone is called a tropic, or TROPHIC, tropic or trophic hormone. They're called both. Book only uses and introduces tropic. This is a tropic hormone, so is this releasing hormone, isn't it? Now, these tropic hormones released by the hypothalamus are called usually releasing and inhibiting hormones. And again, on the same slide, we inhibit this by releasing this inhibiting hormone, get less of this release. So let's follow all the arrows. The hypothalamus releases a releasing hormone, tropic hormone, which causes the anterior pituitary to release its hormone, which causes this endocrine cell to release its hormone to control the target. The level of this hormone is regulated by when you get a lot of it, we have negative feedback on the release of this hormone and negative feedback on the release of that one.
Can you go through that with actual hormones?
We're gonna go through with a whole bunch later on. There's no names up here. Okay? And there are times when the hypothalamus will release an inhibiting hormone that prevents the release of that one. So everybody understand what we're looking at? Hypothalamus releases a tropic hormone called a releasing hormone. Boom. Anterior pituitary releases its hormone, another tropic hormone, which causes our target endocrine cell away from the hypothalamus and the anterior pituitary, and it releases hormone that controls the target. And we almost always have negative feedback on this one and this one. This is how the anterior pituitary is involved. It's an intermediate between the hypothalamus and the target endocrine cell. There's at least 4 or 5 of these that we'll see.
Is it almost like the co-stimulation in a way?
Co-stimulation. No, because those oftentimes happen close together in time. These are separated. They're different. I mean, this releasing hormone has no effect on the target cell. None. It's just this tropic hormone. It's just a series of glands and hormones. The hypothalamus is an endocrine organ, this is an endocrine organ, and that's by definition. 2 hormones, bing, bing, causing and controlling the release of this one hormone that's finally going to produce an effect somewhere in your body. We'll see plenty of them, and most of them will be in chapter 18. Okay? Any questions on it? Get used to this diagram. Make sure you understand it, because we're going to be looking at that and see. So, what's the negative feedback? This hormone on anterior pituitary and the hypothalamus. Almost always both. Okay? And you all understand what negative feedback is. This type of control, negative feedback, will produce what kind of hormone levels?
Chronic secretion?
Chronic, yeah. The high level that's fairly constant. See that? That's the negative feedback I was talking about, producing that kind of control. So, 3 different signals can cause an endocrine cell to release its hormone. We have humoral, neural, and hormonal. Any questions? And again, negative. There can be negative signals. And they can inhibit all. Now, there can be negative feedback and positive feedback controlling the expression of hormone levels. We've already seen the negative feedback, and that's the exact same diagram. There is limited amount, and it's kind of contrived, by the way, in my estimation, positive feedback to where we have of the anterior pituitary release of hormone going to affect the target. And then it has positive feedback on either the anterior pituitary or the hypothalamic hormone. They're not showing the releasing hormone here. And of course, if this has positive feedback instead of negative feedback, you're going to get more of the releasing hormone, more of the stropic hormone, and more of the endocrine. This can't go on very long because you'll very quickly get to an extremely high level of hormone release. So this goes on for a short period of time, and there has to be some mechanism to stop the secretion. Just has to be. Okay? The only example is where we have estrogen being produced that causes, estrogen being produced. That's a lousy example. Forget it. We'll get to it in chapter 28. Okay, estrogen and level of luteinizing hormone, LH. You get ovulation, you get no more estrogen, so then LH stops being produced. But it goes higher and higher levels because of the estrogen. It's not worth explaining here. We'll explain it when we get to there. Okay, hormone receptors and the mechanism of action on target cells. This will take us to the end of the chapter practically. Hormones have to bind, hormones are going to be in the bloodstream. They got to leave the bloodstream, go physically to the cell, bind a receptor somewhere on the cell. They have to bind a receptor, and then this cell is going to have a response. Okay? So the hormone binds the receptor, and it produces a positive, usually positive, effect on the cell. Okay? Those receptors are usually extremely specific for the hormone. The receptor that binds one, there's two components of thyroid hormones, T3 and T4. They both bind the same receptor. Epinephrine and norepinephrine both bind the same receptor. That's rare. Beyond that, there's virtually nothing that has the same kind of response as those two. A drug that binds a receptor and activates is called an agonist. It mimics close enough, and of course the agonist is never found in your body. This is some drug that can be produced that binds the receptor, and of course will produce the effect. An antagonist binds a receptor, but it does not produce a response. So a lot of drugs are agonists and a lot of drugs are antagonists. Okay? Now, this may seem kind of confusing, but certain conditions, other factors affecting the target cell can cause it to put more receptors on its surface or in it, or fewer. If a cell responds to certain signals and puts more receptor, that cell is said to have upregulation. Now, more receptors are going to produce a bigger response in the cell. Fewer receptors will produce a smaller response in the cell. Okay? So, certain conditions in target cells and conditions that are affecting it, let's say, can cause it to put more of these or fewer of those and have a bigger response or a smaller response. That's called up and down regulation. Understand it. We're never going to really deal with it in this course. Yes?
Somebody used downregulation when describing type 2 diabetes. Is that what happens to the cells?
Yes, they're downregulated by definition. They have fewer receptors, so they can't produce a big response. And the glucose stays in the bloodstream. Plenty of insulin, not enough receptors to cause a big effect on the cells. And in that case, for some reason, most of the body cells have downregulated the insulin receptors, and they can't respond, don't respond. And it is a membrane-bound receptor. If you can somehow figure out how to upregulate those receptors, you cure a person of type 2 diabetes. Okay, classes of hormone receptors in the target cells. Remember those lipid-soluble hormones? They go right through the membrane, right through the nucleus, and almost always, there are exceptions, but almost always, that receptor for that lipid-soluble hormone is in the nucleus. There are some that have a receptor in the cytoplasm. Most have them inside. And when the hormone binds to the receptor in the nucleus, then that's going to cause certain genes to be transcribed, translated, modified, move the proteins around, and produces the cellular response. Okay? This is how all of those lipid-soluble hormones work. No receptor in the membrane. They go right through the membrane because they're lipid-soluble, right into the nucleus, and they're almost always, the receptors are in the nucleus. And they're going to cause genes, almost always they're going to cause genes, new genes to be expressed in the cell. Rarely they'll shut down some. Okay? Either way, that's going to produce a slow response. Yeah, it's got to go in here, but you got to take this DNA, you've got to make an RNA, you've got to process the RNA molecule, move it out here, transcribe it, process that, and then the protein can finally have an effect. That's a long sequence of events. Yes?
How long would that take generally?
Hours to a day, easily, to have, till you have a significant response in the cell. A water-soluble hormone binds to a receptor on the surface because it can't go into the cell, okay? Then it's going to cause a change to already existing molecules and can produce a quick response. Sometimes they go through a G protein, activate an enzyme already existing, and then that activates other enzymes that produce the response. So it's a quick response of the cell. Whereas this one will take hours to a day, this can happen in minutes in a receptor cell. Okay? So the difference between these two is just simply the nature of the hormone, lipid soluble and water soluble. Everybody see the difference between these two? Important to understand. So this sums it up, and we'll just go through it. A hormone can bind nuclear receptors if it's lipid soluble. It's going to activate some genes. They synthesize new proteins or enzymes, and then you get the response. Hormone that binds is water soluble, will bind a membrane-bound receptor. It's going to work two ways. They can work through a G protein already existing, and then open or close ion channels or activate existing proteins. You don't have to make the proteins, they're already there. You just have to activate them. Or sometimes those directly, the receptor membrane, the receptor hormone complex activates directly already existing enzymes. And that produces the response. Okay? This is quick, this is slow. Because you got to make new things. Here we're just altering existing proteins. Okay. Now we're going to go through this in detail. Okay, we'll start with the lipid soluble first. The receptor hormone complex, when it forms, is going to bind to some hormone response elements in the DNA. There are molecules associated with certain sequences, and that receptor hormone complex binds to it and causes that gene to be transcribed, and eventually it's going to produce a new protein. There's a delay, a significant delay between that hormone going into the cell and before the cell produces a response. Okay? When that receptor hormone complex is degraded, then no longer will you express this, the proteins. But understand, proteins inside the cells last a while. So, once you make the protein, it's gonna produce a fairly lengthy response inside the cell. It takes a while before the proteins are turned over. Proteins are turned over in the cell, but they last for hours to days. Okay? This is how you turn it off, but understand, once you make the protein, it's there, it's active, it's gotta be turned over, and that takes a while. So it's a delay to get the response, and it lasts a while once it's produced inside a target cell. Water soluble. These happen quickly. Remember, we bind the membrane-bound receptor, and then it's gonna either activate a G protein, or it's gonna directly activate some enzymes. Okay? Almost always, but not always, the cell usually produces an intercellular mediator or second messenger. So you're gonna have the hormone be the first messenger to the cell. It's gonna produce a second soluble entity that then produces the cell response. It's oftentimes cyclic AMP, it's not only, but we're gonna see a whole bunch of examples of that. And if you were to go to the cell and inject the second messenger, you can take a small needle and put it inside the cell, inject whatever that second messenger is, cyclic AMP or whatever it is, the cell will respond. The second messenger has the exact same effect as the hormone. That second messenger is responsible for producing a response inside the cell. And we're gonna see a whole bunch of examples of it. So let's see how, I don't know if this was covered in 189 because it's been a few years since I taught it. Did you cover how G proteins work? A little bit?
Over 10 years or something.
It was a few years ago. We're still gonna go over it.
It was like over 10 years.
When I was a graduate student at the University of Washington, there was this strange fellow, Gallant was his last name, and he was working on G proteins. We thought he was strange. And yet, here we are, talking about his work right here. Okay, so we're gonna have that water-soluble hormone bind a protein receptor in the membrane. The cell put the receptor there. When that hormone binds the receptor, the G protein, which has 3 subunits, alpha, beta, and gamma, okay? The alpha one has a GDP attached to it, guanine diphosphate, and that's why it's called a G protein, the guanine, okay? So, when the hormone binds the receptor, then the G protein will attach to it. And then the GDP is replaced by a GTP after it's bound. Binds the GTP, displaces the GDP. This is all done by changing the conformation of the molecules. After the GTP binds, the G protein separates from the receptor and the alpha subunit separates from the beta and gamma. Okay? Then this is going to produce the response. After a while, a phosphatase is going to come by. It's on the next slides. And this causes the cell response. That produces it. After a while, a phosphatase is going to come by and convert the GTP to GDP, and now we're exactly right back where we were. This combines, and the next time a receptor binds. Remember, after it binds and produces the response, what happens to this? If there's another G protein around, it can activate it. But eventually, what's going to happen to this? Brought inside and degraded. Got to stop the response. Remember, you get a signal to a neuron, you got to get rid of the neurotransmitter. Well, we get rid of the response by, after it produces a few of these, it then is degraded. So, after this one separates, this can still bind another one of those. And this, remember, this G protein, that alpha subunit is the part that produces the response. That's the sequence of events where a G protein is involved in activating a cell and producing a response. So let's go and look at the different ways first that G proteins produce the response. So we're to the point where we have already separated the alpha subunit and we have the GTP. In some cells, it binds to a pre-existing enzyme in the membrane. It's right there. When it binds, it activates this enzyme. In this case is an adenylate cyclase that can take ATP and convert it to cyclic AMP. That's the second messenger. It's not always cyclic AMP, but this is a common sequence of events. Now you can't have one cell have cyclic AMP respond to two or more things, because you gotta use a different. If a cell is gonna respond to multiple hormones, and there are some cells that do, you've got to have different second messengers for those hormones. Oftentimes, it is cyclic AMP. The cyclic AMP is just a messenger. It doesn't produce the response directly. The cyclic AMP oftentimes activates some proteins called protein kinases. A kinase is an enzyme that's going to go to a protein, take an ATP and stick a phosphate group on that protein. And sticking the phosphate group on it activates a protein that produces the response. As long as there's a GTP, it's going to continue having this enzyme produce cyclic AMP. So this one receptor can, say, produce 2 or 3 of these, and each of those can produce hundreds of those. Everybody see that? Because this is an active enzyme, and that's going to activate these enzymes. Those, the cyclic AMP is active until along comes an enzyme called phosphodiesterase that takes cyclic AMP and converts it to AMP. This is not a second messenger, just that version of the molecule is. So this is one way to produce a cellular response from a water-soluble hormone that can't get in. It's a complex sequence of events. Make sure you understand every sequence and part of these diagrams. We're going to go through like 6 or 8 of them. This is just the first one. Any questions? Okay, the next one. Here we have another water-soluble hormone. It happens to be epinephrine in this case. Bind a receptor. We're producing our active G protein. It's going to activate a different enzyme in the cell membrane. In this case, it's called phospholipase C. What does a lipase do? You know. Breaks down lipids. And there's lipids because it's in a phospholipid bilayer. So one of the phospholipids that's in the membrane is phosphoinositol diphosphate, something like that. And this is phospholipids, and that's the phospho, and that's the lipid part. So PIP2 is acted on by this phospholipase C and broken into 2 parts, IP3 and DHE. Both of those, or either, can be the second messenger in the cell. Sometimes both are the second messenger. Other times, only one is the second messenger. Oftentimes, DHE, diacylglycerol or something like that, I can't remember the exact name, activates a protein kinase, and we already saw what protein kinase does. Okay? Or IP3 can be its second messenger, and IP3 is going to cause the release of calcium ions from the endoplasmic reticulum, got muscle on the brain. Release calcium ions, and the calcium ions going to bind calmodulin, and calmodulin's going to produce the response. Oftentimes, this is in a smooth muscle cell. Not always. Okay, so this type of process is going to activate an enzyme, but it's a lipase, uses ready-made, and again, this enzyme can bind, as long as it's activated, because GTP's there, it can find several of them and produce several of these molecules to produce a response. So either one or both of those can be second messengers. Any questions on that one? What's next? Oh, sometimes G proteins open ion channels. So here's a closed ion channel, what is it, a calcium channel in this case? And the hormone binds the receptor. We're gonna activate and use a G protein. The G protein's gonna bind to the channel, open it, ions come in. Now, we know it's gonna change the membrane potential, but that's not what we're trying to get. We're trying to get the calcium ions in, so they bind calmodulin, and calmodulin is gonna produce the response. Okay? When the GTP is removed, you see, this channel is held open by the GTP. When the phosphate takes the phosphate off, the channel closes. Now, eventually the calmodulin is gonna lose its calcium because calcium jumps onto the calmodulin, produces the cellular response, then it jumps off, and some of those times it's gonna jump off and get pumped out. Okay, so what's the second messenger? The calcium ions. Okay, and of course the calcium ions bound to the calmodulin. But if you were to just inject calcium ions into the cell would respond, produce its response that it normally does for that hormone. Okay? It's just calcium bound to calmodulin that produces the effect inside the cell. Some membrane-bound receptors for water-soluble hormones, when they have the hormone bind to it, directly activate an enzyme that's going to produce the response. In this case, directly activates guanylate cyclase, takes a GTP and makes a GMP, a cyclic GMP, which is going to produce the response. And again, a phosphodiesterase inactivates it by converting from cyclic GMP to GMP.
Is that only water soluble?
Yeah, this is always only for water soluble. Everything we're doing now, we finished the lipid soluble with one slide. They all go in and bind hormone response element that's going to cause genes to be transcribed. Rarely genes to be shut off, but almost always genes to be transcribed. Okay? So this is again water soluble, and that's the second message, cyclic GMP. But in this case, we had no G protein involved. The water hormone receptor complex activated the enzyme. There are many examples of each of these because there's a lot of hormones that have been studied. Okay, other receptors, when the hormone binds, they go and phosphorylate proteins that are now active and before they were inactive. And this complex can exist for a while before it's brought in and degraded. And it can sit there and phosphorylate a whole bunch of enzymes, activate them, and then they're activated, produce a response. Phosphodiesterases are present in the cell, and they'll come along and clip off these extra phosphates and inactivate it. So you have no longer expression of, no longer response in the cell. The cells need to respond, then they have to be able to not respond. All you do is remove the phosphates in this case. Don't forget, in every one of these, eventually this is going to be brought inside and degraded. Some receptors directly alter the activity of proteins in the cell. Just the receptor directly activates several protein molecules. Okay? And those altered ones, besides phosphates, it's different than this one, where it's attaching phosphates. They can affect them other ways. cause changes to them and cause them to be activated. Some receptors directly activate proteins in the cell, producing changes, whereas lipid soluble has one diagram, one way it acts. All these water soluble have many ways to produce the cellular response. I would bet there are ones that are known that aren't on that slide. There's still probably other ways. The metabolism is so robust and so many things you can produce responses by that there's probably others out there that aren't listed in our short list of methods of producing a response by a water soluble hormone. But what's common to all of these? What's common to all of these? Membrane-bound receptor. Okay? Receptor always bounds a membrane-bound receptor. After that, we have lots of different ways it can go. Please study each, know exactly how it's working and what the second messenger is, if there is one. Is there a second messenger in this diagram? No. But there is for most. Signal amplification. Now, let's look at water soluble and lipid soluble. The rate of change in the target cell depends on the mode of activation. If it's a nuclear receptor, a lipid soluble hormone, you've got to begin with synthesizing the mRNA, transport it out, process the mRNA, take it out to the cytoplasm. You've got to have it transcribed. Oftentimes you process that protein before you ever get a response, so there's a delay.
2 hours to
Right. Okay, hours to days before you produce a response. But if you're using a membrane-bound receptor, you can have one hormone bound to one receptor. Remember I said each receptor hormone complex can activate one or more G proteins. Each of those G proteins can activate several of those adenylate cyclases. Each of those adenylate cyclases can produce a large number, since those are enzymes, ASE, you can produce a huge number of cyclic AMP. Each of those can activate a whole bunch of already existing. Remember, everything here is already existing. Those G proteins, those enzymes, well, you have to produce the cyclic AMP and these enzymes. They're already present. So you can produce a quick response, and notice it's a cascade because that can produce several, each of those can produce several, each of these can produce a whole bunch, and each of those can activate a large number of even different enzymes. This is called hormone, where is it? Amplification, is it up there? It doesn't say it. It's amplification or a cascade where you get increasing numbers. So this quickly, since everything already exists, this is a quick, and then you end up with a large number. From one molecule, you end up with a large number of activated enzymes inside the cell producing the response. Okay? This can produce a huge response in a cell quickly. What kinds of things do you think you would want to have a quick response to? Name a hormone where you would like to have a quick response to. Insulin, yeah. Blood glucose level has gone up, and insulin, boom, produces a huge response by a water-soluble hormone. What about epinephrine? How quickly would you like to have changes to your physiology when you all of a sudden have epinephrine being secreted in a crisis situation? Yeah. So a lot of hormones have quick responses, so you produce quick, strong responses, water soluble. Ones where we have lipid soluble, you don't need it really quick, and you want it to last a while. So those are for lipid soluble. So see the difference between the two classes? That is the last slide of chapter 17.
Would oxytocin be lipid or water soluble?
You tell me,
I would guess lipid.
what does oxytocin do?
Stimulate
Yes.
So,
To, okay, let's take that. How long do you want that infant to wait? Sometimes they go to sleep and never get fed if we had lipid soluble ones, right? So would you think it's maybe water soluble?
maybe.
What's the other target for oxytocin?
The uterus.
The uterus during
Labor.
labor. Do you want the next contraction coming quickly or wait a few hours? So I would bet, once you study the uses of it, that you would want that one to be water soluble. You want it to be done in a day or two, not several weeks. Okay? So let's think of another hormone and analyze it. We've done epinephrine, we did oxytocin. What about, let's take ADH. You want that quick or fast? Quick. Is it water soluble? Yeah. Okay? If you know the hormones, what it's doing, you can guess what it could be. If you want it to last a long time, it'll be lipid soluble. If you want a quick change, it'll almost always be water soluble. Any others? This is useful to think about. Good question, Bridget. How could you distinguish between neural control and an endocrine response on the basis of response time and duration? We'll take response time first. So, response time for neural or endocrine control? Neural is fast and endocrine is much slower. I mean, action potentials get there pretty quick. On the duration, endocrine lasts longer because even for water soluble ones, they last hours to days, whereas neural control, if you try and have neurons stimulated for a significant period of time, where are they gonna run out of? Neurotransmitter, there's only so much there. I mean, having any kind of neuron fire for a minute is extreme because every action potential that goes down releases more neurotransmitters. They're gonna eventually run out and then can no longer produce an effect. So if you want like the effect to last a long time, use endocrine control. If you would like to have it quick, you'll use neural control. Okay? Are there things that have it both? Probably. But there are things you need to have quick response and last a long time. Such as, name one. We already talked about epinephrine and norepinephrine. It's released quickly by the neurons and then goes into the bloodstream from the adrenal medulla and lasts a while. Not long, but a lot longer than those neurons can fire. That's one where we have both. Are there other examples? I'd have to think about that. Okay? What primary factor determines each cell's hormonal sensitivities? The number of receptors or which receptors it has. Are there cells that could respond to more than one hormone? Yes. In that case, which hormone is affecting it? So think of a cell that can have more than one hormone receptor. There's lots of them. Think of two hormones that could affect the same cell. Well, T3 and T4 are regarded as the same hormone. So thyroid hormones and insulin. could both affect the same cell. And each would have a different method of getting expressed. That's why there's so many. They're both water soluble, both affect virtually every cell in your body. And you have to have different second messengers for those. Chapter 18. I said for a while, these sort of my first, but wait on. So, we're passing in the sign-in sheet in chapter 18 notes. Now, chapter 18, get the notes going around. Chapter 18 covers a lot of hormones and has a lot of detail. I will not cover ones we have already talked to death. And there's a lot of hormones. They're all brought together in this chapter, and you should read them. But so many hormones we have already talked about in detail. Some, we're gonna just gloss over them because I know we're gonna talk about them in a later chapter, specifically the reproductive hormones. So I'll mention them and don't get even to the detail that's here, but we'll talk about them in detail. The book doesn't cover them in those in detail. The reproductive hormones are covered superficially. Okay? That's the only one, the only hormones left to cover beyond this chapter. Okay? So there's a lot of other hormones that we've talked about and we're not gonna cover. So parathyroid hormone, calcitonin, ADH, aldosterone. What other hormones have we covered in detail? ANH. Erythropoietin isn't even mentioned in the chapter, but we've covered it. Okay? So, no, you don't have to go back to the notes. If I ask you a question about those hormones you already know, it'll be only using the material that's in the chapter. And the vast majority, there's rarely anything added, for example, to ADH that we didn't already know, or aldosterone. You already know 99.9% of everything on those hormones. Read over them, but realize it's a review. There's rarely anything added to what you already know. They don't even mention the gastrin and the digestive hormones. They don't even mention them, except indirectly for insulin stimulation, although we covered that too. So, let's go through the learning objectives. And since there are so many hormones, guess what? That means there's a lot of detailed learning objectives. 4 or 5 slides, I can't remember how many. So don't let this startle you. You know a lot of this already. You'd be surprised how much you already know, which is a good thing. Explain the types of information that are necessary to understand endocrine function. Describe the 10 regulatory functions of the endocrine system. I'm not even going to cover that. They're fairly straightforward. They're listed in the first section of the chapter. Please go and look over those. Describe the location and structure of the pituitary gland. You already know that. Explain the physical, neural, and vascular connections between the hypothalamus and the pituitary gland. That's one part that we need to cover in detail and make sure you understand. Describe how the hypothalamus regulates hormone secretion of the pituitary gland. Well, it depends if you're talking about the anterior pituitary or the posterior pituitary. List the hormones produced by the hypothalamus, their structural type, target tissues, and actions. I'm not going to cover that in detail where they cover it in detail, because why discuss these in detail when we're going to go and talk about the anterior pituitary hormones and have to talk about they're stimulated by hormones from the hypothalamus. So that's when I'm going to put it in. List the hormones produced by the anterior pituitary, their structural type, whether they're glycoproteins, lipid hormones, or whatever. Oh, anterior pituitary, they're already always water soluble. Their structural type, so they're going to be proteins, peptides, or amino acid derivatives, target tissues, and actions. Explain the nature of a tropic hormone. That's a hormone that causes an endocrine gland to release its hormone. Describe the conditions that result from over and under secretion of pituitary hormones. That is listed in tables in your book. Make sure you look over those. Describe the structure of the thyroid gland. Explain the processes for the synthesis, secretion, and blood transport of T3, T4. I'll cover that in detail. How does that, why are these big pools of colloid in those follicles in the thyroid? Describe the mechanism of action and the effects of T3 and T4 in the body. Relate how thyroid hormone secretion is regulated. How would you think it would be? Negative feedback. You have constant levels of thyroid hormone. Are there things that can adjust the set point? Yes, but you have a set level. One thing, you would like to go move to northern Canada, your T4, T levels will go up. So you have higher metabolism for the cold climate. I mean, yes, you can move the set point, but it's controlled by negative feedback. Describe the effects of hyposecretion and hypersecretion of thyroid hormones and the pathological conditions that cause the abnormalities. I mean, it's in a table, I'm not gonna go through and look at that in detail. Make sure you do look at those tables. Describe the role of calcitonin in maintaining blood calcium levels and bone health. You already know that. Describe the location and structure of the parathyroid glands. Explain the mechanism of parathyroid hormone action and its effects. Not gonna cover it, we've covered it in this course. Describe the causes and symptoms of hyperparathyroidism and hyperparathyroidism. Relate the location. If you have hypoparathyroidism, what's your calcium level? Low. Hypo, low. And hypersecretion, huh. And then the question isn't what these do, but what do high and low calcium levels do, because that's what it causes. Relate the location of the adrenal glands and explain the embryological origins of the two parts of the glands. Like the posterior pituitary, part of it is neural tissue, the other part is, well, they're slightly different, but the other is not neural tissue. In the pituitary gland, it's epithelial tissue. In the adrenal gland, the cortex, the outer layer, is mesoderm in origin. Describe the mechanisms and actions of the hormones secreted by the adrenal medulla. And that, of course, is looking, of course, at ones we've already looked at before, epinephrine and norepinephrine. Name the layers of the adrenal cortex. You know those from lab. The type of products secreted by each layer, you'll have to learn that. And the predominant hormone of each layer. Okay? Describe the individual target tissues and the responses to the hormones of the adrenal cortex. Explain the role of adrenocorticotropic hormone, ACTH, in the regulation of the adrenal cortex hormone. That is released by anterior pituitary, which causes the adrenal cortex to release its hormones. What do you think regulates the ACTH? A tropic hormone from the hypothalamus. Discuss the causes and symptoms of hyposecretion and hypersecretion of the adrenal cortex hormones. Describe the location and structure of the pancreas. We learned that in chapter 24. Name the hormones secreted by the pancreatic islets and describe their effects on their target tissues. The only two we're going to look at is insulin and glucagon. Explain how pancreatic hormones are regulated. They're not regulated by tropic hormones. They're hormonal, humoral control. Compare and contrast the causes of type 1 and type 2 diabetes. Explain the interactions of insulin, glucagon, cortisol, growth hormone, and epinephrine immediately after a meal and 1 to 2 hours after a meal. What do you think is going to be produced out of that list immediately after a meal? Insulin and immediately and 1 to 2 hours after a meal, glucagon, cortisol, growth hormone. You get the picture. We'll get that in detail. Describe the nervous and hormonal interactions during exercise that will provide enough energy to cells. List and describe the functions of the hormones secreted by the testes and ovaries. We'll just mention them in passing because we're going to get that in our next chapter after 18, which is 28. Explain how the anterior pituitary regulates secretion by the testes and ovaries. The hormones it secretes. Anterior pituitary is a endocrine gland that regulates these two endocrine glands by hormones. We're still not finished. Explain how the placenta acts as a temporary endocrine organ. When it's produced, it produces hormones, so it is an endocrine gland. And we're gonna cover that in chapter 28 and 29, well, actually 29. It produces estrogen, progesterone, relaxin, and probably a couple of other hormones. Very important. List the hormones secreted by the pineal gland and describe their possible functions. Melatonin. Explain the photoperiod and its relationship to pineal gland secretion. It secretes melatonin mostly when? At night. Describe the functions of the hormones secreted by the thymus, digestive system, heart, and kidneys. What's the hormone the thymus secretes? Thymosins. The digestive system, name of 3 hormones. Gastrin, secretin, CCK. There's others. That's just the 3 we looked at. The heart, does it secrete a hormone? ANH or ANP. The kidneys secrete a hormone.
Renin.
Renin is an enzyme, not a hormone. It's an enzyme that does its work in the bloodstream. Aldosterone. No. Aldosterone is secreted in adrenal cortex. Erythropoietin. Give examples of both autocrine and paracrine chemical messengers and describe their functions. Describe the major age-related changes that occur in the endocrine system. We're not omitting any part of this chapter. Remember, I know it seems long, but there's so much of it you already know because we've done all but chapter 28 and 29 up to this point. And I'm not going to cover those parts. So let's begin with the pituitary gland and the hypothalamus. We're just going to get started in this, but this is interesting stuff. The hypothalamus has this infundibulum that supports the pituitary gland. We've got a posterior pituitary and an anterior pituitary. The hypothalamus controls the anterior pituitary because it releases hormones into an area that has capillaries. Those hormones go in the capillaries. They go through a blood vessel that goes to another capillary bed. So the hormones are released here, go into that capillary, don't go back. There's an artery feeding that capillary bed. This blood does not go back to the heart. This capillary bed feeds another capillary bed. So these hormones go directly into those capillaries through that vessel and then go into that capillary bed and diffuse out. If this hypothalamus had to produce hormones that secreted throughout the whole body, you'd have only a few that come back here and could have an effect. But since we have one capillary bed feeding another capillary bed called a portal system, it can produce a small amount that diffuses out right here and produces its effect. The anterior pituitary is controlled by hormones secreted by the hypothalamus into this capillary bed that come out, fuse out here, and affect the cells there. Now, the book gives you a tongue twister name for this portal system, the hypothalamo-hypophyseal portal system. Most people just call it the hypophyseal portal system, just to let you know. But understand, hypo for hypothalamus, thalamo, it's hypothalamus, and then hypophyseal because another name for the anterior pituitary. Anterior pituitary is the neurohypophysis, or the adrenal hypophysis, not neurohypophysis. That's that. Okay. The anterior part's the adenohypophysis. The posterior part, posterior pituitary, is the neurohypophysis.
Together, they're just that
Hypophysis, but I almost always use pituitary gland, posterior pituitary, anterior pituitary. They're lobes. Just speak of them separately. The anterior pituitary is produced from tissue, epithelial tissue from the roof of the mouth during development, and this is produced by actual neural tissue from the brain. Posterior pituitary. I can do it in 45 seconds. There are neurons in nuclei in the hypothalamus, and their axons extend down the posterior pituitary, and their axons store the hormone, ADH and oxytocin. So when these cells receive stimuli, they send an action potential down, release it at their tips, their axon terminals, goes in, the interstitial fluid goes into the capillaries and circulates to your body. So that's how the hypothalamus controls the posterior pituitary. Two different ways, radically different. There's only two hormones. They're called neurohormones, and so are these ones produced up in the, that affect the anterior pituitary, because they're produced by neurons. I usually just call them hormones. So I usually say that ADH is a hormone, not a neural, although it is released by a neuron in its more proper term. If it goes in the bloodstream, I just call it a hormone, and it circulates to everything. And I will see you next Tuesday. What happens